In this video, I am going to discuss about Ramachandran plot, which is a very important aspect of protein 3D structures. Ramachandran plot gives us information about that which type of secondary structure of protein is possible and which kind of secondary structure of protein is not possible. So it will be giving us the information about torsion angles, then how the proteins are arranging themselves in the secondary structures, and what is the position of different secondary structures on the plot or on the Ramachandran plot. So before going ahead, we just see what is the correct definition of the secondary structure. Then we will be looking at different points and different concepts related to Ramachandran plot. So the definition of secondary structure is that secondary structure is the local spatial arrangement of a polypeptide's backbone atoms without regard to the conformation of its side chains. Means secondary structure is not considering the conformation of the R group of amino acids, or the secondary structure is not considering the structural aspects of the side chains or the conformations of side chain. It is just showing us, but it is just using the backbone. The backbone of protein is containing alpha carbon, then peptide bond, then alpha carbon, then peptide bond and all. So a structure which is dealing with, or which is the combination of arrangement of the backbone atoms, is called as secondary structure, where it is not considering the conformation of its side chains. Now, to go ahead, we just have to learn little about the peptide bond. So we are having a polypeptide here. In this polypeptide, we are having the peptide bond, that is NHCO bond. So we are having bond between nitrogen and carbon, and this bond is called as peptide bond. Now just near to this nitrogen we are having the alpha carbon. So here we are having alpha carbon, then peptide bond, then again alpha carbon, then peptide bond, then again alpha carbon, then peptide bond, and so on. So here we can see that on the alpha carbon three things are attached: the previous carbon double bond oxygen, the next NH, an R group, that is the side chain group, and the hydrogen. Okay, so in this structure I have not shown the hydrogen, it is the self understood part. Now we look at the structure of the peptide bond and we see what is happening. So when we look at the peptide bond, that is C double bond ONH, the nitrogen is containing the lone pair. The lone pair single bond double bond resonance can be there. Then these lone pair can shift here and this double bond can shift to oxygen. So we have another structure where we find double bond between carbon and nitrogen. So because of resonance, the peptide bond is having partial double character, and because of this partial double bond character, it is becoming rigid and it is difficult to twist. So this reason, this peptide bond is difficult to rotate or it is difficult to twist because of its rigidity, and that rigidity is coming because of partial double bond character, which is caused by resonance. Now if we cannot rotate it, then what we can rotate? We can rotate these bonds, right? So because of this rotation, the system can be occurring in two parts, cis and trans. So what is trans? That one R group which is present on the left of the peptide bond is having a transposition relative to next R group. So they are in opposite direction, which is providing a kind of stability. And what is cis? Where both the R groups attached to the peptide bonds are in the same direction. So cis is less stable in comparison to trans. So in the whole peptide sequence, we are finding that R group down, then R group up, then R group down, then R group up, then R group down. So they are arranging themselves in a transposition to get as much as stability in the structure. So from this slide, what we have learned, what is the alpha carbon, then what is the peptide bond, then why the peptide bond is rigid, and the system is present in a trans way, it is not present in a cis way, because trans way is having more stability than the cis conformation. Now we move ahead and we see how we are rotating the things. So again, in this structure, this is our alpha carbon, okay, this is our peptide bond, and this is our next alpha carbon. So we'll be looking at here only. So since the peptide bond is a planar bond, because it is not rotating, it is present in a same plane, so this thing cannot be rotated. So what we can rotate? We can rotate this bond or this angle, and we can rotate this angle or this bond. So we are having first angle, which is called as psi. So what is the psi angle? Psi angle is between the alpha carbon and peptide bond carbon. So the bond between peptide bond carbon and alpha carbon is making an angle, a torsion angle, which is called as psi, which can be rotated. Now what is the second angle? The second angle is phi. So what is phi? Phi is the angle between alpha carbon and the nitrogen of the peptide bond. So we are having two torsional angles, phi and psi. Phi is between alpha carbon and nitrogen of the peptide bond, and psi is the angle between alpha carbon and the carbon of the peptide bond. So now you can rotate this bond, and you can rotate them to have minimum steric repulsion, or you can say minimum trouble in stability, and by rotating this bond you can achieve the most stable structures, which we will be seeing in our Ramachandran plot. Now we try to understand how we are rotating the things. So this is again the same structure. This is the alpha carbon. This will be the psi angle between alpha carbon and carbon of peptide bond. This will be phi angle between the alpha carbon and nitrogen of the peptide bond. So in first condition, we are just rotating the psi angle. So psi angle is flipped. So again, it was a straight structure of psi angle. Now it is rotated structure of psi angle. Now again a straight structure. Now this time we will be rotating the phi angle. This is rotated structure of phi angle, and this is the straight structure. So we can see that psi and phi angles can rotate themselves from -180 to +180. Means they can rotate themselves to a 360 spectrum, or to a total angle of 360 degrees. So the values of the psi and phi will be -180, then 0, then +80. So the range is -180 to +180. Again we see, so here is our psi angle and this is our phi angle. So the psi angle is rotating, then again it is becoming straight, then phi angle is rotating, and again it is becoming the straight angle. So we can rotate the phi and psi angle to decrease the steric repulsion between different groups, to make the structure or secondary structure more stable, and the values of psi and phi are -180 to +180. Now we again see this kind of rotation in detail. So here we are having the psi angle 0. So this is the alpha carbon, this one, then the carbon behind it is the carbon of peptide bond. So they are making which angle? The psi angle, the angle between alpha carbon and the carbon of the peptide bond. So right now the value of psi is 0. You just rotate it to 180. So now you can see the carbon double bond oxygen has flipped upwards. So this was the condition where psi was 0. Now this is the condition where psi is 180 degree. So this is positive rotation, so it is +180. We can do negative rotation also, that will be -180. So I can say that the value of psi is between -180 degree to +180 degree. Now we see the same kind of rotation for the phi angles. So the phi angle is between the alpha carbon and nitrogen of the peptide bond. Okay, so right now the phi is 0, so they are not having any angular difference. You just rotate it, so you will find that the whole group has rotated, and now the value of phi is 180 degree. So that is again positive rotation. We can go in negative direction, so I can say that value of phi will also be from -180 to +180 degree. So right now we have understood about two torsion angles, psi, which is the angle between the alpha carbon and carbon of peptide bond, and phi, which is the angle between alpha carbon and nitrogen of the peptide bond. And we have looked different rotations by looking at different structures, and we are finding that they are having the values between minus 180 to plus 180 degree. Now different secondary structures will be having different values of these angles phi and psi to keep the minimum steric repulsion to increase the stability to a maximum. So the graph between the values of phi and psi is called as Ramachandran plot. So Ramachandran plot is nothing but on x-axis we are having the values of phi from minus 180 to plus 180, and on y-axis we are having the values of psi from minus 180 to plus 180. Now we have divided this graph into four quadrants. Quadrant number 1 is having the negative values for both phi and psi. Quadrant number 2 is having positive value for psi but negative value for phi. Quadrant number 3 is having positive value for phi as well as positive value for psi, and quadrant number 4 is showing the positive value for phi but negative value of psi. So these four quadrants are divided based on the values of phi and psi, and on x-axis we are keeping phi and then y-axis we are keeping psi. Now the pink color in this graph is showing those values of phi and psi angles where the secondary structure of protein is not possible. What do you mean by that? It is not possible means at those values or at these values of phi and psi we are having higher or very high steric repulsion, and because of that high steric repulsion the structures are not possible to attain. The white regions are showing that in these angles or in these values of angle different secondary structures are possible. So in first quadrant we are having alpha. Alpha means the right-handed alpha helix. So for alpha I can say this is the region where the alpha secondary structure is possible. So I can say the alpha helix is having the value of phi in negative region as well as the value of psi in negative region. Now we are having second structure, that is P. So P is coming in this region, that is showing parallel beta sheets. So for parallel beta sheets we are having positive psi and negative phi. Then A is anti-parallel beta sheet, it's nearly coming in the same region. Then T is twisted beta sheets. Then C is collagen triple helix. So these structures, all of them are having positive values of psi and negative values of phi. Finally, L is left-handed alpha helix, which is having positive value of phi as well as positive value for psi also. So in this Ramachandran plot we have learned that different structures or different secondary structures are having different set of phi and psi values, and these different set of phi and psi values are decided based on their stability. So proteins are folding themselves in a secondary structure by rotating the torsion angles phi and psi in a way where we are having least steric repulsion. Now one interesting observation I tell you. So the right-handed alpha helix and left-handed alpha helix are mirror image to each other. So this is the point of origin. So this point of origin can be said as point of inversion. So at this point the structures are inverting themselves. So the right-handed structure is converting into left-handed structure. So the white regions again are the possible reasons for the value of phi and psi where the protein secondary structure can exist, and pink areas are showing the reasons or the values of phi and psi where the secondary structure of protein cannot exist. Now we take two special cases for the Ramachandran plot, and the first case is a peptide which is made up of glycine. So glycine is having you can say R group as hydrogen. So the side chain is very flexible, so it is having so many possible reasons or so many possible values of phi and psi for in the Ramachandran plot. So all these dots are showing okay, that in this region the glycine, pure glycine peptides can arrange themselves in secondary structures because of the over flexibility of side chain R group, which is nothing but hydrogen. Since the proline is a very rigid amino acid, it is actually not an amino acid, it is an amino acid. In that case, because of high rigidity, it is having less values of phi and psi, where the secondary structure for the proline peptides can occur. So normally these are different reasons, and so many different reasons can be possible for glycine because of over flexibility, and very less number of reasons can be possible for proline because of its rigidity. So again, what is the crux? The crux is the planar system is present in the peptide bond. It is rigid, so it cannot rotate itself. So there are two torsion angles which can be rotated. One is psi and one is phi, and when the protein secondary structures are forming, they are keeping the values of phi and psi in such a way where they are keeping the steric repulsion at least, or you can say very less value of steric repulsion. So we are having different values of phi and psi for different secondary structure. So for beta sheets or beta strand, it is the value of phi is between minus 150 to minus 100, and value of psi is plus 120 to plus 160. For alpha helix, minus 70 to minus 60 and minus 50 to minus 40. For 3 by 10 helix, it is minus 70 to minus 60 and minus 30 to minus 10. For polypeptide second helix, it is minus 80 to minus 60 and plus 130 to plus 160. So I hope you understood the basics of Ramachandran plot and why you are requiring these rotations and what exactly the Ramachandran plot is telling you. So thank you guys. Thank you very much.